Hello everyone, welcome to our channel. So today we are going to discuss about what is Kaplan-Meier survival plot, how we can plot the KM plot using our program. Okay, so KM plot basically gives an idea about the survival probability of a particular gene in any cancer condition, whether the high expression or the low expression of that particular gene is prone to the survival probability of a patient. So let's go to the topic in detail. So before going to generate our KM plot, we need to have two types of data sets. One is tissue expression data set in any cancer condition. The second one is the survival related information about the genes of interest. Okay, let's see how we can identify or find such two data sets. So the first database that we are going to use is UCSC Sena portal. So that provide tissue expression data set. So you have to click on this, the first web page, and go for launch Sena. And from here, so it will take some time to load. Okay, from here we can go with this data set, and you can select either any of the tissue expression data set. So I am going with the breast cancer GDC TCG breast cancer tissue, and you can download this star TPM expression data set. Okay, so you can see that the unit of the transcription is log2 TPM plus 1 value, and you can click here to download the data set. Okay, so that is going to download here. So I already download the file, so I'm going to close this. Now, if you want to get the survival related information, you can go to this portal called CBio portal, which provide all the clinical information about different kinds of cancer. So I'm going to take the breast cancer here again, and I am going to select the GDC TCG file. Yeah, we can select this one, invasive breast carcinoma TCGA GDC file. Here you can see the sample size, and you can go with this explore selected studies option, and you can click here to download the file. So now we have two files. Now let's see how we can filter and clear the data before going for the KM plot analysis. All right, so your files have been downloaded. So let's see what are the files that we have. So this file contains information about sample IDs, patient IDs, etc. So let's do one thing. We can copy this file, and we can open an Excel file format. So I'm using WPS here. So let's see what are the data that we have. Okay, so I'm going to take only some information. So we can have the sample ID here, and when you scroll here, scroll on the right side, you will get the information about the survival relationship. Yeah, exactly. So you can select this one also. All right, so now we have sample IDs, overall survival in month, and overall survival status. So this is one file, so you have to rename this file. The second file that we downloaded is the tissue expression data set. So we have to unzip the file, and you can go with the WinRAR option here. We can extract here. So this will give you the tissue expression data sets. As you can see here on the first column, we have ensemble IDs, and followed by further columns the tissue patient sample IDs. Okay, so before going with our data, we have to do one thing. We have to, we have to convert this ensemble ID into gene official, like GNC gene official symbols. So for that, I will be using DB to DB conversion tool, or you can use any conversion tool. So let's see after converting these ensemble IDs, how the data will be. Okay. All right. So we have two files here. One is a survival information related file here, and second one the tissue expression data set. So here I converted the ensemble IDs to corresponding gene symbols. Now we have to clear some data here. For example, here we have the sample IDs and overall survival status. So I have to make it as a binary digit. So I just need 0 and 1 only. So we can do one thing. So we can remove this leaving option here. So we can control H here, and I am going to replace this file with blank. So all the 0 leaving will be replaced by 0. Now you can do the same thing. So I'm selecting this one and just spacing here and just replacing the space. Okay, that is done. Now let's see whether the data are clear. Okay, now we have some issues. So we have this not available information. So you can do one thing, you can delete this file. All right, and let's see whether any NA files are present here. You can search here NA. All right, only one file is remaining. So I'm going to delete this one also. Now we can sort the file, custom sort, and you can go with sample IDs values. Yeah, that's it. Now you can save this file. Now around 1000 files, 1000 tissue samples are there. You can save this file. Okay, and here we have around 1226 sample IDs. So we just need to select only the common sample IDs present in here. Even if you don't take the common one, what happens is that so the files or the tissue sample IDs present here will only be converted to their values in order to identify or in order to get the KMPRO. So before that, it will be better if if you filter the or find the common tissue sample IDs. So we can do one thing. We can select this, copy, and we can open a new Excel sheet and we can transpose this one. All right. So I'm just transposing this one and I am having this sample IDs here. You can do one thing. You can see the common among this one. So I'm going to do one thing. So I'm just going to open this Venny tool. It is used to identify the common values, intersection values. I'm going to paste here. So we have, how many we have? It's not fetching it. Yeah, we have around 1103 values are there. I'm going to take this one also. All right. So okay, there is one problem. As you can see that here we have some extra values. So the A is being added to this 01A. So we have to replace this also. So you can do one thing. So we can select this one and we can replace this one with 01. All the values are replaced, and now we can check with this. Okay, there are some other values. 11A is the okay, so we can do one thing. 11A can also be replaced. Oh, sorry, not here. Okay, we can delete this, replace. All the values are replaced. Now let's see whether this worked or not. Okay, I'm just pasting here. Okay, exactly. So it's working now. Now we have some more values here and some more values here. Okay, we have 01B and 11B 6A left. So we just need to replace this 11B. So 11B is going to be replaced with 11 and we have 06A which is replaced by 06. All right, and one more thing is that what is that 1B. Okay, 01B is going to be replaced by 01. Now again we can go with the finding the common ones. Okay, so yeah, okay, all right, so again we have 1100 samples here, and these I don't think so even if you replace any values it won't be same. So what we can do, we have to select only this 11,000 samples here. All right, so instead of going for any program, we can do one thing. We have to select the samples from here. So I'm going to copy this whole file here and open a new Excel sheet. I'm just going to paste here, deleting this first one. I'm just taking these values and maybe I can take the first 500 here. Okay, I'm just going to take the first 500 values. Okay, yeah, I'm going to take this 500. Okay, there is 499. Okay, I'm just copying these values. What I'm going to do is that I'm going to transpose this file here. Okay, and I'm going to select this one and take it in Notepad. All right, so just open a new file here and just paste it here. As you can see that there are this space, so this is not a single space. You have to select the space, control A, or maybe copy this one and control C, press that one, and you can select the whole string and control H, and you can paste this. As you can see that I think it's a tab or maybe a couple of spaces, and you have to replace this by single space. Why? Because I will show you why. Okay, that is replace. If you want to see the change, you can again copy this one, control A, control S, and paste. Yeah, here, so the tab space is being replaced by single space, so we don't need to replace further. So control A and control C. Now what I am going to do, I'm going to select only, okay, before that we can delete this, row, and maybe the selection will be lost. Okay, control A, control C, and I'm going to filter here. So if you give a space, a single space between two values, it will be identified, the value. So we have 499 values separated by single space, so it will be identified. So that is the benefit of WPS. Okay, yeah, so all the values are identified, and if you have doubt, you can see this one. Okay, 510 values are selected, which means there are some duplicate. There can be some duplicate values or some common values. Let's see whether we have duplicates or not. Don't worry, we are going to do one thing. I'm going to select the complete data sets here, and maybe I can open a new Excel sheet and paste it. Okay, now what we have, I think, again some 500 values are there, right? Where did I paste it? Okay, somewhere I have pasted, right? Yeah, exactly here. So 499 selected, 500 is 500. So I have to select from this 500, again copy. The same procedure, I'm going to transpose this one, copy here, spacing here, and selecting the space here, control A, control H, control V, and give a single space, replace all this file, again control A, control C, and we can go with, yeah, this is the one, this file, and I'm going to select again this one. Now let me see how many will be there. 602. All right, fine. So I'm just taking all these values, just pasting here. All right, so we were supposed to have 1100 samples here, but we have 1113 samples. So 13 samples are more, or 12 samples are more basically. So one is actually heading. So let me see whether any duplicate values are there. All right, so just go to data here, data, home, conditional formatting, highlight cells rules, duplicate values. Yeah, okay. Now let me see whether any duplicate values are there. Custom sorting, symbols, cell color, this one, exactly. So this is what happens. So we have basically duplicate values. These many duplicate values are there. We have to delete that. So these are the sample ID duplicates. What we have to do actually, so we have to, so if you have two values or three values, we have to find the mean of that. So we have to find the average. So let me see whether we have only duplicate values here. So I can do one thing. I can again go for custom sort. I can go with symbol, and values are sorted by alphabetical order. Okay, let's see. These are same values, same, sorry, same, these are same, these are same, all are same. So we have around 11 exactly 11 samples are duplicate. So we have totally 12, and if you remove the duplicate values, it will be 11. So we have around, how many we had, 11,012 samples are there, around 11, sorry, 1112 samples are there. So if you remove the 11 samples, so we will be having 1100, around 1100 samples. So what we have to do, we have to do one thing. We have to give space here. Sorry, not here. Just give space here. Okay, do one thing. So we need to give space here, and we have to give space here. Space, 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 space. All right. So why we are going to space here? Because we are going to find the average of these things. All right. So I'm just going to find the average. So let's see how we can find. So it's a simple thing. So what we have to do, we have to give the option here, average, and we have to select this one, comma, this one, right? Yeah. So the average, so this is 5.9 and 3.9. So the sum is 8.9. So the average will be around this one. Yeah, it will be the same. So you have to drag here until the last file. So you can do the same with all of this file. So let's take some time. I will finish this one and I will show you what happened next. Okay, now I have identified all the average of these duplicate values, right? So these are the values here. I just want to remove these values and I have to save this one. So before that, I just want to copy this here, right? So I'm just want to replace this blank file with the name of the sample. Just want to do this for all the files here, and then we can delete the duplicate ones. Okay, so be patient, I'm going to finish this soon. All right, one more to go, sorry, two more to go, and yeah, now it's been finished. So we have to replace these values here. So just want to replace these values, just selecting this and copy and just replace these values with this option values here. And now we have to save these values, the third one, these values we have to save. So what we have to do, we have to delete this one, we have to delete this one, and we have to delete this, yeah, right, delete this, delete this. All right, it's exactly, yeah, exactly, yeah. So this one, this one, this one, this one, this one, and this one. So we have to delete all these values here. Okay, now our data is clean. So we found the mean average of the TPMs here. Now let's see. Yeah, so we have 1100 samples here. Now we can go with the venue again here. So I'm just clearing all these options here. So let me see. Copy here. I'm copying this one. Just pasting here. And one more file we have. Yeah, we have this file here. Just copy, and where is this file? This file. Right. So one sample is there. So okay, it's because maybe 111C, but let me see whether it's because of AODB. There is no AODB, so we don't need to select this one. So we can remove the sample because it's not present in both of the data sets. So we can do one thing. We can delete this sample here. So even though our sample space is high, so we don't need to rely on this one. All right, so this we can delete this one. Right, now we have 1100 samples here. So just rename this file, and now we are good to go with the R code, and we can fetch the game plot. All right, so what I have done, so I have this file here. So I have taken all these values and transposed it into a previous format. So all the tissue sample IDs here as the first row or the column names. So this is our tissue expression data sets. I just renamed this file here, and I have the survival related information here with overall survival status as binary values. Okay, now we can have our program here. So I already have the code here, and you can give your uh, the pwd here, sorry. Okay, so your location of the files here. So I just created one folder in download section, and I have two data here. One is survival data, so I just renamed with this file, and we have one expression data set as well as. Okay, and I'm going to find the survival curve km plot of these three genes. Okay, so these are the three genes. I'm going to identify the expression in breast cancer, breast invasive carcinoma, and so you can give the color code and the size, font type, everything in this one. So if you want, I can provide you this R code with the comment or the description, and I'm going to make this as a PDF result. All right, I'm going to select all this code here and just go in with this run option. Okay, the file is running. Yeah, the result file is being generated. Now let's see the result of this. Okay, here we have the file. Okay, now we generated three Kaplan-Meier survival curve for CKS2 gene and MIR3671 gene PRKCB. So this is the KM result of our three genes, and I'll give you some basic information regarding that. So we will be giving a detailed description or detailing about how we can analyze a particular Kaplan-Meier plot in the next video. Before that, I can give you something. So these are the p-values here. So this one is significant. Basically, this is not significant because we are checking the p-values, taking the p-values 0.05 less than 0.05 will be more significant. As you can see that these two values are not 0.05, and we have one more ratio here. This is HR ratio, hazardous ratio, we can say that. So hazardous ratio that gives how the expression of any particular gene is prone to the survival probability of patient. For example, here in this one, so the hazardous ratio is 1.5, which means if it is greater than 1, that means the higher expression in the sense the red line, so those patient with higher expression of this particular gene will be prone to high death rate, though the survival probability will be less, as you can see from the figure itself, right? So the red line or the high expression line is going down, so this is why it's happening here. And here also you can see that it's greater than 1.5, so the higher expression will be going down. So the expression, high expression of PRKCB can cause to less survival probability or more death chance. And here we have 0.74, which is indicating that the low expression of this particular gene is prone to less survival probability. Okay, this is just a reverse. If it is less than 1, the low expression is prone to death. Low expression of the CKS2 is prone to death, and if the hazardous ratio is greater than 1, the higher expression values are prone to death. Okay, so we can discuss in detail about what is Kaplan-Meier survival plot, how we can analyze, what are the things that we have to look into, right? All these things will be covered in the next video. I hope all of you understand the basics of Kaplan-Meier plot, how we can do it. If you have any doubt, just put it in the comment section. We can give detailed analysis about that. And if you have any query, if you want me to improve anything, please let me know. Okay, thank you for watching.